Hey guys, I know I sped through the end of the last lecture really fast, so I just want to review just very quickly. We talked about plate movement, the tectonic plates moving, how you have geographical isolation because you are moving continents further away from each other. You're forming new environments, you're losing old environments, and because of this, the environments are changing, and so does life. So, an example would be the flora and fauna, so the plants and animals that you see in Australia are really unique because Australia separated really early, and then remained isolated for 50 million years, and so the life that you see in Australia is very different than the life you see in other parts of the world. We talked about mass extinctions. We said this is the sudden loss of many species in geological time. It could be caused by asteroid hits or just other disasters on Earth. There are 5 major extinction events. Some of these you're familiar with, some of them maybe you're less familiar with, but here you have them highlighted. Um, so the red is the total extinction rate, and so then the blue is going to be the number of families that were lost. One of the things that you should be able to see is that the number of families is kind of like where things are. Number of families in blue is kind of increasing. And over time, but you do have these sort of really high peaks in the total extinction rate, so the families that are lost. So the Permian extinction was about 250 million years ago, and you lost 90% of the species. The Cretaceous extinction was one that most people have heard about. This was 65 million years ago, and this is the loss of the dinosaurs. And so that's why I say it's the one that most people have heard of. There's really good evidence that this event was caused by an asteroid that hit the Yucatan, which then caused a nuclear winter. So what happens because of mass extinctions? Again, we've got climate changes. You have areas that are open for the surviving species to exploit. You're going to have usually a really rapid period of speciation due to adaptive radiation. And you also have many new species that are formed in just a very short period of time. So your lecture question from the last lecture was, in your own words, why are mass extinctions important for evolution? And so you should really kind of go back and look at this last slide and kind of look at adaptive radiation and what happens because so many areas are open for exploitation after a mass extinction. Now, what I want to focus on today is evolutionary novelty. And so what do I mean by that? I'm talking about new body forms. So where do those new body forms come from? So we're going to be talking about evolution and development, and essentially how you can have very slight genetic divergences that can be responsible for producing very major body form changes. Two examples. The first, heterochrony. This is going to be the change in the timing of developmental events or growth rates. And then you also have what we call homeotic genes or Hox genes. And these are genes that are responsible for changes in spatial patterns. So an example for heterochrony is different growth rates in humans compared to that of a chimpanzee. So if you look down here at the chimpanzee fetal skull compared to the human, what you should notice is that there's a lot of similarity. So early on, humans and chimpanzees tend to develop at a very similar rate. However, that changes once you start looking at older chimps and humans. So actually pretty quick after birth, you'll start to see changes in how the human and chimpanzee skulls develop. So the human skull is going to continue to grow in different places, and you can kind of see the difference in size in the brain case versus the jaw. And so again, chimpanzee and human fetal skulls look very similar, have very similar growth rates that ends soon after birth, and you can see the differences in the shape and size of a chimpanzee skull versus a human skull at the adult stage. Now we're going to look at Hox genes. So here we're looking at just at the very top a hypothetical vertebrate ancestor. So this is an invertebrate with one Hox gene cluster. So this is a cluster of genes up at the top. Then you have a hypothetical early vertebrate. And so now it is a vertebrate and it has two Hox genes clusters. And then at the bottom, now you have a vertebrate with jaws that has 4 Hox clusters. And so what happened is each time the Hox genes have been duplicated, and so it's changing the development of these organisms. There are a couple specific examples. You have the expression of Hox gene 7 in UBX for the insect body plan. You also have PIDX1 in stickleback fish. So here you've got the Hox gene 7 with the UBX and just the different body types. And then we're looking at the stickleback fish. So we had two hypotheses. So hypothesis A was looking at differences in the actual coding sequence of the PIDX1 gene. What we found out was that the 283 amino acids in the PIDX1 protein are identical. And what we're doing is we're actually comparing two different types of stickleback fish. So we've got the marine and then we have the lake. With hypothesis B, here you're looking at differences in the regulation of expression of the PIDX1 gene. And so it was shown that the PIDX1 gene is expressed in the ventral spine and mouth regions of developing marine sticklebacks, but it's only expressed in the mouth region of developing lake sticklebacks. And so that was sort of the big difference, is that where the gene is being expressed. So we talked a lot about natural selection and how the environment selects for certain traits within a population. We want to take a second and talk about species selection. And so when we talk about speciation we're looking at the species level now. Speciation is equivalent to the birth of a species, and extinction would essentially be the death of a species. And so species that leave the most new species are going to be the most successful. And so this encourages branches in the tree of life. And so as a species changes over time and it starts to evolve, and you start to see these changes in gene and allele frequency, and you end up having maybe one species that branches into three different ones because of migration or sympatric speciation because the environment has changed or there's a new geographic feature. The most successful species are going to be the ones that are able to branch out into new ones and evolve as the environment changes. So your summary for this chapter, make sure you guys understand the steps to chemical evolution. You want to be able to recognize the use and limits of fossils. Again, we've been doing this for several chapters now. You should know the key events in Earth's history, what happens to evolution and mass extinction, and then again some examples of evolution and development. So your lecture question for today, how can Hox genes affect development, or how can changes in Hox genes affect development? So again, shoot me a message via the class website and answer that question in your own words. Thanks, guys.